Hey learners, welcome and welcome back to the part 3 of this chapter, Cell the Unit of Life. In this lecture, we will talk about eukaryotic cells. We are already done by their few of the topics like plasma membrane and their structure, their functions. Now in this lecture, we will talk about the cytoplasm and their endomembrane system. So the topics to be covered in this chapter are ER, GC, lysosome and vacuoles. We will mainly focus on these topics first. So let's begin by the chapter. First we have Golgi complex, which is a conceptual term of cell biology that refers to a functional and structural series of three important organelles in the pathways, where G stands for GolgiBody, ER stands for Endoplasmic Reticulum and L stands for Lysosome. While each of these membranous organelles is distinct in their terms of structure and functions, but these are still considered together as an endomembrane system because of their functions are coordinated with each other. Since the functions of mitochondria, chloroplast, peroxisome are not coordinated with the above components, so these are not considered as the part of the endomembrane system. Now this is the proper diagram of the endomembrane system, where we will see these three of the components like endoplasmic reticulum, Golgi complex and lysosome vacuoles. They all lies in the common part of the cell, which is called as the cytoplasm. You all know what cytoplasm is, right? They are present between the nucleus and the cell membrane. And the cytoplasm can be divided into two main parts, which are called as cytosol and tropoplasm. Cytosol, which is the liquid matrix of the cytoplasm. You already know these terms from class 9th, that their liquid is dense, viscous in nature. And the tropoplasm is something which is solid part of that cytoplasm, consists of living organelles, which we will discuss in this chapter, and other non-living organelles like inclusion bodies that we already know, right? Gas vacuoles, dictyoplasm and many of such. So other than that, in the endomembrane system, where we will see the synthesis and secretion of molecules like proteins and lipids throughout these organelles, and at the end we'll combine all these in a common diagram. So let's begin by the endoplasmic reticulum first. So this is the diagram of endoplasmic reticulum, which is used as ER as an abbreviation. It's a large complex organelle found in the eukaryotic cells. In the electron microscope studies of eukaryotic cells, it reveals the presence of a network of reticulum of tiny tubular structures which is scattered in the cytoplasm, that is called as the ER, endoplasmic reticulum. And if we talk about its membranous structure, it's a single membrane organelle which continues with the outer membrane of the nuclear envelope, as you can see in the image how they are directly connected with the RER. It is visible under the electron microscope because of its branched and network-like structure, which occupies a lot of huge part of it, which is almostly about 30 to 40% part of its volume. It's a huge portion, isn't it? Now you know why it is important for the cell functions. Functions like it is enough to give cell a definite mechanical support and many of such things. We'll discuss them later on. And this ER divides the intercellular space into two main compartments, which are luminal, which is inside the ER, and the another one is extraluminal, which is about the cytoplasm. Now they are arranged in three distinct components, which are cisternae, vesicles and tubules. We will see them. Cisternae are long, flattened and unbranched unit of structures which are arranged in the stacks like pattern. Then we have tubules. These are the irregular, branched or membrane bounded tubules which may remain free in the cytoplasm, or also they can be associated with the cisternae. And lastly, we have vesicles, which are very simple, oval, single membrane bound structure. Now suppose if the ER is a tree, then cisternae is its broad branches and big leaves, which functions as a site where the substances like proteins are produced and stored, where the tree manufactures and stores its food, and the tubules are like the thin branches or the vein of the leaves, you can say, It helps to connect the cisternae together to transport the materials internally. They act like the pipelines connecting different parts of the ER networks together. And then the vesicles are like the fruits or seeds of that plant that drops and travels throughout it. They are like the delivery system which carries the tree's product to its destination, like to the other plants and like the other animals. Similarly, they carry proteins or lipids from ER to GolgiBody or out of the cell also. Now there are two main types of endoplasmic reticulum. One is rough endoplasmic reticulum or RER, and another is smooth endoplasmic reticulum or SER. So we will talk about them. Let's begin by RER first. See, the only difference between rough ER and smooth ER is because of one organelle which is called ribosome. We'll see them. So in the rough ER, it is covered with the ribosomes on its outer surface, which gives it a more rough appearance as the name suggests. And if you talk about its membranous structure, it continues with its outer membrane of the nuclear envelope, and it is made up of more cisternal flattened like structures as compared with the tubules. Its main function is protein production and its secretion. See, it has ribosomes on its structure for protein production, and once the proteins are formed, these are proceeded and folded into the ER lumen by ensuring its proper shape. And soon after that, these proteins are transported throughout the cell, especially when these are secreted out of the cell, and they can be inserted into the cell membrane, and also they are used in the lysosomes. They can be also used by Golgi apparatus via the transport vesicles. See, RER mainly involved in the protein production, not in the modification. It is done by the GolgiBody, to be more specific. But once upon a time, this question was asked in the exam, that they modified the protein into glycoprotein, and they asked their roles. While the RER is mainly for the protein synthesis, but also does some modifications, while the GolgiBody performs further more complex modification. So you can go with the GolgiBody, but when it is not given in the option, you can mark RER also. In that case, it will be also right. Then we have smooth endoplasmic reticulum. It has no ribosome on its structure, which gives it a smooth like appearance, where it gets its name smooth endoplasmic reticulum. They are continuous network like structure which is often extended from the RER, and they have more tubules as compared to the cisternae. They function as the site for lipid synthesis like phospholipid for cell membranes, cholesterol and steroid hormones, especially in the liver and adrenal glands. Then these cholesterols is converted into the steroid hormones via specific enzymes which are present in the SER, and these steroid hormones are lipid soluble in nature, so they can diffuse directly out of the cell without the use of any vesicles or any other such organelles. For example, we have adrenal cortex in the cortisol and aldosterons, and ovaries in the estrogens and progesterones, and testosterone in the testes. So all of these enzymes are derived from the cholesterols only. They act like the perfumes that spread quickly throughout the air, similar to the bloodstreams. And the lipids like phospholipids, triglyceride and cholesterol, these are present in the liver cells and the cell membranes of the different cell organelles. And they also function as the detoxifying agent for the drugs and other harmful chemicals, especially in the hepatocyte liver cells. Their membrane contain this special cytochrome P450 detoxifying enzymes in them. These enzymes modify these harmful toxic substances out of the body, and their main goal to do all this is to make these lipid soluble toxic substances water soluble, so they can be excreted out via urine or bile, and then it sends these clean filtered detoxified product into the blood streams. By this process, it removes all the toxic chemicals, drugs, alcoholic and metabolic waste out of the body, and protect the cell from the poisoning agents. And it is also become essential for our body. Without it, even small amount of toxics could be deadliest for us. Now suppose we'll take an example of a drug addict person who takes alcohol heavily so that his SER level increases and can't be able to handle more detoxification work, and his body start oxidizing these alcoholic compounds into acetaldehyde, which is a toxic intermediate compound, and these acetaldehyde is more toxic than the alcohol itself. It can damage the cell membrane, protein and DNA, and in response to it your hangover symptoms and the liver inflammations, and can kill your liver cells and leads to the fatty liver issues. And the long term alcohol use increases their SER enzyme, and after a certain level of time their SER becomes so much developed that they don't get affected by 1 or 2 bottles of alcohol, so they start taking more alcohol to feel the same effect, that they detoxify it at the same time and the body don't feel clogged. On the other hand, a new drinker feels its effect quickly. Some sort of 1 or 2 drops or glass of alcohol can easily raise their blood level, while the regular drinker need more alcohol to feel the same, and that they don't get affected by 1 or 2 bottles of alcohol, so they start taking more alcohol to feel the same effect. And that is the reason why people says every time you have to take an increased repetitive dose of alcohol, that is more harmful to your body. Now imagine a scenario where you are taking bad drugs like any of these alcohol, chitta, or you at the same time you are also taking some good drugs like any antibiotics. Now what will happen here? See, your liver is so much developed that it will detoxify that medicine along with that drug even before the medicine do their work. That makes sense, right? Why many of these antibiotics doesn't work on the drug addict persons. Now it is in your hands. Do you still want to be that alcoholic sit, or then be ready for the long term effects? Okay, now let me ask you one question. On the basis of your understanding you have to answer me. So the question is, which cell has more rough endoplasmic content in it? And these are all your options. You have to choose between all of these. You can take your time. Now the answer is very obvious, the protein producing cells, and that is correct also, but they will try to fool you in the exams by giving you options like proteinaceous hormones and lipid hormones. So don't be confused, always cross check your answer. See, since we all know that all the enzymes are proteins, almost 99% of enzymes are proteins, but not all the proteins have the same origin or the same pathways as of the enzymes, right? So you have to choose between them. Either it's a protein synthesizing enzyme or lipid synthesizing enzymes, because the steroids hormone are lipid based and made from the cholesterols only, and they required SER for their synthesis, while the proteinaceous hormones are made by the ribosomes and they required RER, which is a protein based enzyme. And the systolic enzymes, they are also protein based, but they are made by the free ribosomes in the cystoles, or you can say the cytoplasm of the cell, not by the fixed ribosomes of RER. So the correct answer is B, protein based hormones. Now let's move by the next slide. Then we have the another cell organelle which is called as GolgiBody, Golgi apparatus, or you can also say Golgi complex. It's a single membrane organelle discovered by the Camillo Golgi first in 1898. He was an Italian scientist, was working on the nerve cells of an owl by using some of his own staining techniques, which are used to stain some certain cell structures in the dark color, by which the invisible parts of the cell can be observed even under the lighter microscopes. So while he was studying the nerve cells, he noticed that there is a network of dark stained membranous structure is there near the nucleus, and this later on this was named as the Golgi body, and it was named after him as an honor, although it was a very controversial topic at that time. Many of the scientists were used to think that the Camillo Golgi experiment is just an optical illusion which is caused by the staining, but later on, by the discovery of electron microscope in 90s, it was confirmed that it is nothing but a real or complete structure of the cell organelle. That is interesting, right? Okay, now if we talk about their structure, the Golgi body is made up of three different parts, which you already know their names. Okay, but we'll see them. First is cisternae, then we have tubules and vesicles, but they are mainly composed of cisternae. You can see in the image they are the flat disc shaped sac like structure that comes with the diameter of 0.5 to 1 micrometer. You can see in the image that many of the cisternae are concentrically arranged in the stack-like structure which are parallel to each other, while the number of cisternae depend on the need of that cell, which varies among different animal cells. Now you can see there are two different phases of the cisternae. One is the convex surface of cisternae, which is towards the nucleus, which is called as the cis phase, or you can also say the forming phase. And the another one is the concave surface of the cisternae, which is towards the membranous structure, which is also called as the transphase or maturing phase. While both of these cis and transphase of these organelles are entirely different from each other, but still they interconnected with each other. Then these tubules are the branched and irregular tube-like structure which are associated with the cisternae, which you know that, right? And then we have vesicles at the end. They are working as the transition vesicles and mature vesicles. We will see them in detail. Now these are few of the functions principally performed by the Golgi body, which are packaging material, then modification, and end by the sorting. So we will see them. Let's see how they do so. So we will see step by step packaging of material into the cell. So first the proteins and the lipids made in the endoplasmic reticulum arrives in the small transportation vesicles. Then these vesicles fuse with that cis phase with the receiving site of the Golgi body. Now you know why this Golgi body remains in the close association with the endoplasmic reticulum or nucleus. Okay, fantastic. So when these vesicles fused with the cisternae of the Golgi body, it starts its modification, which is a step 2, where the sugar are added to the protein or lipid structure to form glycoproteins or glycolipids, and also there were some phosphate groups are attached to it and some sulfate groups are attached to it. So once they ensure their correctly folding shapes, these modified molecules are packed into the membrane bound vesicles and are moved towards the trans phase or maturing phase, which is the sipping side of the Golgi body. You know, when you order some product on some e-commercial websites, these products are customized in the workshop before shipping to your address. So it is similar to that only. And at the end, sorting of the Golgi body takes place where they decide where each product should go to their target. Like some of the vesicles who carries the digestive enzymes, they become the lysosomes later on. And some of the vesicles that fuses with the cell membrane and release the contacts out of the cell as a secreting material by the process of exocytosis, and some other vesicles that move towards the cell membrane and merge by adding new proteins and lipids to the plasma membrane. Now you know how these number of proteins which are synthesized by the ribosomes on the endoplasmic reticulum, how they are modified in the cisternae of the Golgi body before releasing it from the transphase. And these always remember that this Golgi body is the most important site of formation of these glycoproteins and glycolipids. And beside its main jobs like modifying, sorting, packaging of proteins and lipids, the Golgi body had some several sort of extra roles which you might not find always in your textbooks. We will see them here. One of these roles is the formation of acrosome in the sperm cells. See, in the human sperm cells, the Golgi body produces the acrosomes, which contains the enzymes needed for the egg penetration. You already know that in the previous classes, and if not, then we will discuss it in reproduction chapter. So during the fertilization, how it occurs there. and also it acts as the pigment granule production, where it helps in the formation of pigment containing vesicles like the melanin granules in the skin cells, and also forms the cell wall material in the plant cells by synthesizing pectin, hemicellulose, and other polysaccharides for building their plant cell wall. You know that, right? Then we have the lysosomes. These are the single membrane bound vascular structures formed by the process of packaging in the Golgi apparatus. Yes, lysosome forms from the vesicles, but not just the any vesicle. These special vesicles that contain the enzymes synthesized in the rough endoplasmic reticulum and then transported to the Golgi apparatus in the small transport vesicles. And at the end, these vesicles become mature into functional lysosomes. It is usually oval or spherical in shape. Other than its shape, lysosome is a very classic example of polymorphic cell organelle, where poly means many and morphic means its morphological structure, where the lysosomes don't look the same all the time because they can exist in different forms. They can change their shape and sizes during their life cycle. And these isolated lysosomal vesicles have been found to be very rich in almost all the types of hydrolytic enzymes that works actively in the acidic pH of lysosomes. Now don't tell me if you are unaware of it. Do you? Okay, let me take this in a brief. You know what hydrolysis is, right? Hydrolysis is the breakdown of any molecule or biomolecule in the presence of water, whereas hydrolytic enzymes is a biological catalyst that speed up that hydrolysis reaction even more faster by breaking down these biomolecules like bigger molecules as a protein, fats, and nucleic acid polysaccharides into smaller units by adding water to their bonds, which is optimally active at the acidic pH of 5, which is comparatively low as compared to the outer pH of the cytoplasm, which is in neutral pH of 7.2, where these hydrolysis enzyme break down these biomolecules like proteins into proteases and further into amino acids. Similarly, lipases break the fats into glycerol and fatty acid, while the nucleases break the DNA or RNA molecules into nucleotides, and glycosidases breaks the polysaccharides into simple sugars, and further they are distributed to all over the body. Don't forget all of these enzymes like proteases, lipases, nucleases, and glycosidases all are active only in the acidic pH. Now, can you imagine without these enzymes, the hydrolysis of the big molecules can be very slow. Sometimes it takes days, weeks, or even up to years at the normal cellular condition. But with a hydrolytic enzyme, the same reactions can happen in seconds or even in the milliseconds, in the blink of your eye. Isn't it interesting to see one take days to hydrolyze a reaction and another do it within seconds? I mean, can you see? The enzyme makes the reaction 1,000 times faster than the previous one. Isn't it crazy? Now, can you tell me why is this the huge speed differences there? I'm sure you know the answer, right? Yes, because of the enzymes. But what does this enzyme do here? The enzymes lower the activation energy, the energy that is needed to break the bonds and to stabilize the transition state so that the reaction moves faster and faster. I have already explained these topics via the graph in the previous chapter, so I am hoping that you are aware of all these things. Anyways, now we will see their structure in a little more detailed way. So let's begin by the next slide. So now we have the structure of lysosome, and if you look by this sorry and if you look by this diagram of of lysosome, which is about zero point 0.2 to zero point 0.5 micrometer in their size, with a very basic pH range of 7.2 in their cytosol. Now take a look what's inside the lysosome. So it contains a number of acid hydrolase enzymes that are active under the acidic condition. Their names are given as such: nucleases, proteases, glycosidases, lipases, phosphates, sulfates, and phospholipases. Other than these 5 to 6 enzymes, there were about 50 to 60 more different types of digestive enzymes are there, including these, where each enzyme is specialized for the breakdown of a particular type of biomolecules, but together they all work for the intracellular digestion, which works under the acidic pH of 4 to 5, which is more acidic than the pH of the cytoplasm. So how do lysosomes still stay acidic and able to maintain that pH in it? So to do that, the membranes of lysosome has an active H plus pump protein in their structures to produce the acidic pH in their lumen of lysosomes. So this H plus ATPase protein pump, it's similar to that of water pump in your house, but instead of water it pumps the hydrogen ions or the proteins into the lysosome. This pumping requires the energy from ATP as it is a pump protein against their concentration gradient. You already know this. It is one of the active transportation system by the last lecture, right? So they continue the pumping of H plus ions inside the lysosomes to maintain the right environment for the acid hydrolases enzymes to work efficiently in it. You can see their structure in the image, how H plus pump mechanism works. So without the help of these special proteinaceous structures, it is kind of impossible to maintain that acidic pH in the lysosomes, and as a result the lysosome couldn't be able to perform the vital roles like digestions and recyclings. So it would be a complete loss for the whole cell. So yeah, it is that important. Okay, so now we have discussed enough about the structures of lysosome. Now we will move by the different types and forms of the lysosome. Okay, so let's see the next slide. Okay, so now let me clear you that this slide is completely optional. Either you want to do it or not, but it is not given in your NCERT. So you can skip to the next part. So these are the few main types of lysosome. One is the primary lysosome, then we have secondary lysosome, and then we will see the residual bodies. So let's see them. Primary lysosome or the storage granules, these are the small membrane bound vesicles which are freshly formed from the GolgiBody. As these are the newly formed lysosomes, they stores only the acid hydrolase enzymes in the inactive form, although they are not yet involved in any kind of digestions. Then we have secondary lysosome or heterophagosomes, which are also called as digestive vacuoles. Now these lysosomes are formed by the fusion of primary lysosome and a phagosome. So now what is phagosome? These are the vesicles that contain external materials that could be anything like food, vacuole, or any waste that gives it a larger and irregular shape. Now the enzymes are active at this stage and perform functions like heterophagy, where it digests the materials taken in from outside the cell. For example, in the macrophages, the secondary lysosomes digest the engulfed bacteria. Lysosomes are abundantly found in the phagocytic cells, which I will explain in their functions. Then we have residual body, and please, I'm sorry for the background, there were some puja going on. Can't do anything with that is also important for your spiritual growth. So yeah, for now you'll focus on the screen. So the residual body or the tertiary lysosome, these are the final stage in lysosomal activity. Once the digestion is completed, all over the leftover indigested materials remains inside these lysosomes. These lysosomes are often expelled out of the cell by exocytosis, or sometimes they remain inside the cell, especially in the long living cells like neurons. But they are mainly eliminated out of the cell because they contain waste that cannot be further digested anymore. They are like the trash bags that lifts after the cleaning process. And yeah, before going to the next slide, we have one more thing. Other than heterophagosomes, there is one more autophagosomes, or you can say autophasic lysosomes. It also acts in the digestion, but not of the foods, but the other dead old cell organelles, which are fused by the primary lysosomes. So let's see this in more detail in the next slide. So these are few of the functions of lysosome. We know that these lysosomes are mainly involved in the digestion of food particle or any other matter. So we will see by the different types of digestions performed by the lysosomes. So first we have the intracellular digestion, where we will see heterophagy and autophagy respectively. We have already seen these terms in the last slide. In this we will discuss their main objectives with the diagrams. So the heterophagy lysosome forms when a lysosome fuses with a vesicle containing material. That material could be anything. It could be any foreign particle, any food material, or any bacteria also. So that material brought into the cell by the process of endocytosis, phagocytosis and pinocytosis respectively. So before doing that, we will see what are phagocytic cells or what is phagocytes. So it is a type of cell that engulfs and digests the foreign particles or any bacteria or any kind of cellular waste. It plays a very crucial role in the body's complete immune defense system. These cells are more like the body's cleanup crew, which removes all the harmful substances by contributing to the overall immune response to our body. In the blood, fluid and circulation chapter, you will see these topics in a more detailed way. Other than that, we have endocytosis and pinocytosis. Pinocytosis is itself a type of endocytosis only, where the cell absorbs the liquid dissolvable substance from the surrounding. You know these terms already from the previous chapter, right? Okay, now let's come to the point. So these enzymes inside the lysosome starts break down that foreign material into it. For example, we have macrophage cells. You may have heard their names before. So they actually engulfs that bacteria and by the phagocytosis, and then the lysosome digests them inside it. The main purpose for doing all this is defense, to destroy all these pathogens if there is any, and the nutrition is one of their functions for the digestion of ingested foods. Then we have autophagy, which is also called the self-eating of the cell. These are the structures formed when the primary lysosome fuses with a vesicle containing cell or any of its old or dead damaged cell organelle to recycle their components. It is like a recycling unit inside the cell. And remember, autophagy takes place during the cell starvation only. See, the cell starvation means a condition where a cell doesn't get enough of the nutrients or energy supply like glucose, amino acid, any fatty acid, that starts triggering the autophagy for its nutritional requirement to recycle the old organelles, and if the prolonged starvation can even lead to the cell death after a period of time. And for example, we have like if there is any old mitochondria that could be broken down to reuse its amino acids and lipids and spread it to the cell so that the cell can survive even during the starvation by these recycled nutrients of that old mitochondria, and the damaged part are removed out of the cell, which are no longer used, which doesn't have any nutritional value in it. So these unused parts are removed out of the cell to keep the internal environment of the cell healthy. Now I hope it is clear now. You can see on the screen there is a beautiful diagram given there on the comparison between autophagy and heterophagy side by side, showing how lysosome helps in both the process. Now let me know if you if there is any doubt. It's better to take a shot to review it real quick. So let's see. It's a well-defined 3D structure of lysosome given on the screen. So on the left side, autophagy is given, which is self-eating, and on the right side, phagocytosis is written there, which is a mode of heterophagy, which is other eating. Now look on the straight left side of this partition, where you will find a damaged mitochondria, or you can imagine any other old organelle. It is not mitochondria always, it is up to you, you can imagine them. So it is, the mitochondria is targeted. A double membrane structure forms around it, which creates a autophagosome structure. That autophagosome fuses with a lysosome later on, and the enzymes inside the lysosome that breaks down the mitochondria into simple molecules, like molecules into amino acids, into sugars, into fatty acids, and many more. Then these molecules are recycled and reused by the cell, especially useful during the starvation of the cell. And this is an intracellular digestion which digests internal components in it. Now take a look on the right side of this image, where a bacterium or any food particle, bacterium to be more specific as it is written there. So a bacterium comes from outside the cell. The cell engulfs it by the phagocytosis process, forming a phagosome, which is a vesicle that contains the bacterium inside it. Now the phagosome fuses with a lysosome, and this forms a phagolysosome later on, and the lysosome's enzyme destroys that bacteria into the simple harmless substances out of it. And most importantly, it is an extracellular digestion inside the cell. It means it digests the materials that enters the cell from outside the environment. So in short, in both the autophagy and heterophagy, the lysosomes provide the enzymes that do the actual breakdown in them. They are the common digestive machinery for the both of the process. By this simple thing, house, you can remember it for long period of time. Autophagy, where the cell cleans its own house like damaged organelles, and in the heterophagy the cell cleans the outside mess like the bacteria and such things, where they both rely on the lysosomes for their digestion. This is common for both the processes. Okay, now we have the extracellular digestion. See, normally lysosomes do intracellular digestion which is inside the cell, but only in some cases they also performs the extracellular digestion, where they secrete their hydrolytic enzyme outside the cell to break down the substances into the surrounding. For example, I just don't want to confuse you by the example, right? So just first let's take a scenario which you can relate. Okay, so come on, tell me one thing, that have you ever wondered how our bones repair themselves after a fracture? Do any one of you want to tell me? Yes, the calcium carbonate can be used in the fracture treatment because it has the ability to promote the bone regeneration and its growth. But that is true, but that's not what I am asking about. We have a very special cell here which is called the osteoclast cell. They are the specialized bone cells whose main job is to break down the bone tissue, not inside the cell but outside of it. Similarly, not just in the case of fractures but also in the bone growth and remolding, and also in the maintaining calcium balance in the blood. During osteogenesis, the lysosomes of osteoclast cell, which is a bone-eating cell, as I said, they dissolves the unwanted part of the bone with it by performing the extracellular digestion of bone by releasing lysosomal enzymes into the lacuna, which is a space beneath their osteoclast cell. By this extracellular lysosomal digestion performed to clear away their damaged bone during a fracture. Without this cleanup, your bone would never heal properly. And once that old bone is cleared away, the another type of cell called the osteoblast cell comes into that bone's matrix, and this is how the bone remolding and fracture healing occurs in that cycle. As you can see in the image on the left side, you can see a fractured bone is there. At this stage, there is a damaged bone tissue that need to be removed before the healing can start it. And in the right side of the screen you can see there is osteoblast cell in the orange color, Once the osteoclast removes all the dead bones, then the osteoblast cell moves in to secrete new bone matrix into it so that the new bone can form in it. This is like a first digestion then construction kind of process where digestion done by osteoclast cell and construction occurs by the osteoblast cell. Yeah, that it is. And their extracellular digestion also occurs by the fungal lysosomes. As I said, the lysosomes in the osteoclast cell, they secrete their enzyme outside the cell to digest their bone matrix and release the calcium and phosphate into the blood, similar to that, but in fungi only. So let's just move by it. Okay, the next cellular digestion is also called the autolysis and autodissolution. Understood? Yes, if you understood, then well and good. If not, then I will repeat the same topics in this tissue chapter. So yeah, no worries, work better by the next time. Okay, so let's move by the third type of digestion, which is called the autolysis, or also called as the autodissolution. So let's see them. Did you know your cells have their own recycling system, or you can say their own suicidal buttons? Interesting, right? So let's talk about their cellular digestion versus autolysis, the two very distinct roles played by the same lysosome. So let's see them. Where the cellular digestion is all about the normal life processes, where you will see the self eating and others eating processes, and in the autolysis it's about the cell death mode. So the cellular digestion are of two types, intracellular digestion and extracellular digestion, which we are already done by. Now we will look what is autolysis. Autolysis is the self digestion. See, the auto itself means self and the lysis means the breakdown or digestion. Now let's see their examples along with their diagrams. Now think of it, what happens when the cell is badly injured or no longer needed anymore? That is when the lysosome switch from being a helpful cleaner to a self-destructive button by its own self. This is called the autolysis or autodissolution. And the lysosome burst out and release all their powerful enzymes into the cytoplasm so that the cell ended up there digesting itself completely. It sounds scary, but let me take you through the examples. For example, the tails of a tadpole larva disappears during the metamorphosis by the process of autolysis, where the autolysis of tail occurs. And in the humans, after their childbirth, the uterine line shrinks back to the normal size with the help of this process autolysis. Not just these two examples only, but any old cells can be destroyed by this process only. Wait. That is why lysosome called as the suicidal bags of the cell. Because when their enzymes are uncontrolled, the very first thing they can destroy is their own cell. Yes, that's true. Let's see how they do so. So in the normal condition, their lysosome is in a stable condition, right? But once their lysosomal membrane ruptures under any certain condition like cell injury by any toxic chemical, radiation, mechanical damage, or by any infection or starvations of the cell, its membrane becomes unstable. Usually when one or two lysosome ruptures, nothing serious happens to the cell. Let me explain why. Since the cytoplasm has a near neutral pH of 7.2, which is more or less very far from the acidic pH of lysosome, because it is active only at the pH of 5, which inactivates most of the leaked enzyme out of it. So the enzymes from a single lysosome are usually harmless in the cytoplasm. Because of the low concentration of the enzyme, a single enzyme's lysosome contains only a tiny fraction of the total enzyme pool that neutralizes the enzyme's effect before they can cause any damage. But when too many lysosomes press at a time, the cell literally commits suicide. That is why it is called as the suicidal bags of the cell. Then we have the last cell organelle of our endomembrane system, that is vacuole. So let's see them. Here is the diagram of the vacuole. Now it's a membrane bound sac like structure which is present in the cytoplasm of the cells. It is filled with water like fluid that contains enzymes, sometimes iron, sometimes sugars and organic molecules, and sometimes other pigments or maybe toxins also. and other excretory products. So we call water plus all these minerals together as sap. So water plus mineral is sap. It has many other materials in it which are not useful for the cell. That is why we call it a storage and a waste management unit of the cell. It works like similar to that of. If you look by their structure, the vacuole is bounded by a single membrane structure which is called tonoplast in the plant cells. Actually, it's a plant cell character only. They are barely found in the animal cells. So these are the structure of plant cell, animal cell and amoeba, which shows the structure of vacuoles in each one of them, where their size and functions vary depending on their type of the cell accordingly. Okay, so let's see them. First begin by the plant cells. In the plant cells, the vacuoles can occupy up to 90% of the volume of the cell. If you look by their functions in the plant cell, the tonoplast facilitates the transport of a number of ions and other materials against their concentration gradient to the vacuoles. Hence, their concentration is significantly higher in the vacuole than compared with the cytoplasm. Look, you note my words. Lysosome and vacuoles. These two organelles are very special in terms of their structure, as both of them have the special channel proteins in their structure for the transportation of molecules against their concentration gradient. Let's see their functions in a little detail now. So water storage is a main role of their function, where the vacuoles hold large amount of water in their cell sap, which maintains their turgor pressure, which keeps the cell rigid and support the plant structure also. Then it also stores some important inorganic ions in it, such as potassium, calcium or chlorine ions. They are stored in the vacuoles, and they can be released out in the cellular processes and in any significant need of the cell. While being in the vacuoles, they helps to maintain the ionic balance inside it, and also helps to regulate the osmotic balance and enzyme activity. They helps to maintain the cell metabolism and pH regulations also. Then we have pigments. So the vacuoles store many pigments in it, like anthocyanin, which acts like a pH indicator, which depending on their pH it shows red or pink color in the acidic condition, purple color in the neutral condition, and also turns blue in the alkaline conditions. And it can also be colorless in the very alkaline conditions sometimes. These pigments gives bright color to the flower and fruits for pollination and seed dispersion. This question has been once asked in the NEET exam, that which water soluble pigment is found in the cell sap and vacuole. Now you know the answer, that is anthocyanin. Perfect. And as I said, they act like the cellular dustbin, so they stores the metabolic byproduct and many other waste in them. And other than that, they also acts like the toxic substances. They store the toxic substances like the alkaloids, for example nicotine in tobacco, or they also store bitter compounds in them to protect the cell from self poisoning, and also for the defense mechanism, like to protect the plant from the herbivorous animals. So these are 4 to 5 roles of the vacuoles in the plant cell. Now we will move by the vacuoles in the amoeba. So let's see. So the vacuoles can't be same all the time, right? They vary according to their types, as I said, and they also vary according to their roles which they play. So other than that of the sap vacuole, we have few more different types of vacuoles. So let's see them. Vacuoles are not just the characteristic of multicellular, but they are also in the unicellular organisms such as protozoa. For example, you will find that in the amoeba, paramecium. So let's see them. First we have the food vacuoles, and yeah, food vacuoles are in both protozoa and in the some other animal cells. Their main function is the digestion of food. So let's see this with this example, how amoeba uses these food vacuoles to digest and engulf their particles. So this diagram shows the formation and function of a food vacuole during the phagocytosis in a cell. And the cell could be anyone like amoeba, or can also be a white blood cell. So let's assume it amoeba as it is written there. For now, look how the cell extends its plasma membrane around a food particle by the process of phagocytosis called the cell eating. That food particle could be anything, bacteria or any organic matter. So the food particle gets trapped inside the cell. In the second diagram, the formation of food vacuole is given, where you can see once the food particle is engulfed, it gets enclosed in a membrane-bound sac-like structure inside the cytoplasm. This sac is called as a food vacuole, or you can also say phagosome. So it temporarily stores the ingested food material in it. So in the third diagram, the fusion of lysosome with the food vacuole is given there. So where the small vesicle containing digestive enzymes in it, after fusion, the enzyme enters in the food vacuole and the vacuole is now called as the phagolysosome. In the fourth diagram, you can see the digestion of food particles in it, how the lysosomal enzyme break down the food into the simple nutritionals like amino acid, into sugars, into fatty acid molecules. And these nutrients are further passes into the cytoplasm to be used for the energy and growth requirements. The undigested remaining waste materials remains in the vacuole only. And at the end, all of the waste material is expelled out from the cell by the process of exocytosis. Now you know how food vacuole works. Good. Okay, now let's see the another vacuole we have. Now we have the contractile vacuole. They are again in the protozoas like amoeba in the paramecium also. So they are the characteristics of the freshwater protozoa only. Contractile. The word itself means the ability to produce the contraction. So it performs the function of osmoregulation where it prevents the cell from bursting out by continuously collecting the excess water entering from the cell by the osmosis and the pump it out. So let's see this by the diagram. I know this diagram is not pretty clear but good enough to understand, right? So let's do this. As you can see, there is an amoeba which lives under the fresh water. Now suppose this is a freshwater screen which is hypotonic in nature. It means the more water is outside on the screen and less solute is inside the amoeba. Due to the osmosis, water constantly enters the cell. So if the excess water is not removed out of the cell, then the cell would swell out and even can burst out of the pressure. In the second step, you can see the formation of the contractile vacuole occurs where the inside the cytoplasm the small vesicles collect excess water in it, and these vesicles gradually fuses together to form a large contractile vacuole in it. Have you noticed in the diagram there is something dotted structure surrounded outside the vacuole? That is the mitochondria. They provides the ATP energy for pumping the water to the vacuole. Filling of the contractile vacuole takes place in the third step. You can see there where the vacuoles slowly fills up with water collected from the cytoplasm and it keeps growing in size like a balloon filled out of the air. And in the step 5, actually there is no step 5 after it. The cell repeats this contractile vacuole cycle. As the water keeps entering in the cell, the vacuoles keep forming and filling and then contractiling again and again after. This ensures that the amoeba survives without bursting. And at the end, this contractile vacuole act like a water pump in the amoeba, right? It protects the cell from bursting. It also maintains the osmotic balance in it, and without it the amoeba, it is impossible for the amoeba to survive in the fresh water. Amoeba would die without it. So this is the diagram which beautifully explains how the osmosis works using resin by the Kismis in Hindi. So if any of you students who are still confused, then look carefully this time. We are familiar with resin, right? We eat it every day in our home, and we also put it in puddings. It tastes delicious, right? So this dry fruit comes with a very little water inside it, but with a very high sugar concentration, right? Their cells of the resin are plasmolized, means they are shrinked because of the all of the water lost during the drying process. Now in the second step, we will take a beaker filled with plain water in it. We'll soak the resin in the beaker. So the water in the beaker has the higher concentration as it is a hypotonic solution. Now we'll compare to the inside of it. And in the inside of the resin, it is fully concentrated with the sugar. Now due to the osmosis, the water molecules will move from outside to the inside of the cell, means from the higher concentration to the lower concentration through the selectively permeable membrane of the resin cells. And in the step 3, you can see there are hollow round resins in the watch glass are kept there, because after some time the resins absorbs all the water from the beaker and swells up. The cells become turgid full of water by the endoosmosis. Resins are the perfect model to demonstrate endoosmosis, correct? Okay. Now the resins, as I said, they are the analogous to the amoeba. Just like the resin swells up in the water due to osmosis, similar to the amoeba in fresh water, they also swells as the water enters in it. But the amoeba prevents bursting with its contractile vacuoles, which resins don't have. Now tell me why the contractile vacuoles are mainly found in the freshwater protozoas only. Yes, because of the osmosis, that is correct. But why they are not in the marine amoebas? Do you know the reason? Tell me. Because of the fresh water, which is hypotonic in nature, so that the water from outside the amoeba has a lower concentration of solute compared with the inside the cell. This makes the water continuously move into the cell by the osmosis to keep the amoeba safe. While in the marine water, the amoebas or the marine organisms, they don't have any of such organelle in them, because they don't face this constant influx of water in it, so they don't need any contractile vacuoles. As you can see in the diagram, I have attached it for your clarity on the left side. There is no contractile vacuole in the marine water amoeba, while the freshwater amoeba has a one. If nothing controlled this thing, then the amoeba would swell up and burst just like an overview soaked resin. I hope it is clear now. Let's conclude this together. The contractile vacuole is an adaptation to life in the freshwater protozoas, which helps the amoeba to maintain osmoregulation and to avoid bursting. Now we are done by the food and contractile vacuoles. They were given in your NCERT. Now we have a few more different types of vacuoles, like the gas vacuole and storage vacuole. I'm sure you are already aware of these kind of vacuoles. So, but still, let's take a look on them. So the gas vacuoles, they are present in the cyanobacteria and some other prokaryotic cells. They are mostly found in the aquatic cells, like the photosynthetic bacteria, or in the blue-green algae, or in the purple and green bacteria. If we talk about their structure, the gas vacuoles, they are the protein bounded structure. Unlike the other vacuoles, they don't have a lipid membrane, but they are made up of a protein cell that is permeable in nature to the gases, as it has gas filled in its structure but not to the water. Its main function is to regulate the buoyancy. Actually, buoyancy is a term in physics. I hope you know that, buoyancy or thrust, where it's an upward force which is extracted by a fluid that opposes the weight of an object. It is like the phenomena. It's basically where that allows the object to float or rise in the fluid or liquid or gases also. So Let's come back to the point. The gas vacuoles helps the bacteria to float or sink in the water by allowing them to reach the depth where the light intensity and the oxygens are optimal for their photosynthesis. So they reaches on the top of the water level for the water and oxygen for the basic life survival processes. It is like a survival strategy for the cyanobacteria. By adjusting the buoyancy, they can avoid harmful lights at the surface, or even they can move deeper during the harsh condition. While they can also use these gas vacuoles to position themselves in water so that they can get the right amount of sunlight. So they can use it in any ways. They are like the life jacket in the water for these aquatic bacteria, which help them to float at a particular right depth for their survival and to perform such important life functions. So as you can see in the image, I have attached one of their image on the screen. These gas vacuoles are made up of many small subunits called the gas vesicles, where each gas vesicles is a tiny shape that they are made up of proteins, and they are rigid enough to resist the collapsing under water pressure. Can't you see in the image how they avoid collapsing? You can imagine by your own. Other than all this, there are some other storage vacuoles in the plants that are specialized vacuoles for storing proteins and lipids or starch into it, which are common in the seeds to provide them nutrients during their germination phase. So yeah, that is all about the vacuole. Unlike the plant cells, where vacuoles are larger and more permanent in their size, shape, but in the humans and animals, there is no such vacuoles are there. If there is any vacuole, then it must be smaller temporarily and only forms when as needed during the process like the endocytosis and exocytosis. They forms for their food transportation all around it, and they fuses with some other organelle. That is all about the vacuoles in animals and humans. So yeah, we are done by this chapter. Let's conclude what we saw in this chapter. The biogenesis of endoplasmic reticulum, GolgiBody, lysosome and vacuoles by the gull complex, where endoplasmic reticulum is the production unit of the cell, GolgiBody is the packaging and quality control unit of the cell, and lysosome is like the recycling and disposable department of that cell. And from the next time, once you think about a resin swollen in the water, always remember that is the osmosis. Okay. So yeah, that is it for today. And I'm sure that nobody will explain you in such dynamic way even on the internet. So yeah, no offense, although so take advantage of it. Okay. In the next lecture, we'll move to the mitochondria. Till then, keep learning, keep watching, and subscribe for more biology lectures.